So there we are. So welcome to Cardiff University and particularly the Cardiff University Otter Project. It's a really exciting day because we're going to be performing an autopsy on an otter. I've been dying to get in here. It's brilliant. So I'm joined by Emma and Laura and also by Principal Investigator Liz Chadwick. Liz, how are you?
I'm good, thank you, Reece.
Fantastic. I am so interested in this project. So tell me more about the Cardiff University Otter Project. What's it all about? How did it get started?
So it started about 25 years ago. At the time there'd been a serious decline in otter populations, and so there's a real conservation drive behind the project. And so we aimed to collect any otters that were found dead anywhere in England or Wales at the time. We've now expanded to Scotland.
Wow.
And doing a post-mortem examination to really try and find out all that we could about them, not just how they died, but also whatever we can find about how they lived.
So that's really important because a lot of people watching at home today will assume that we're just dissecting an otter for the sake of dissecting an otter and filming it, but actually this is part of a really important piece of research, undergoing research, which is going on here at Cardiff University.
Yeah, that's right. All sorts of interesting information can come from the post-mortem, right from the basic biology to the genetics to contaminants, all sorts of things.
So what can I expect today? I'm in here, I'm very, very excited. Give me an idea of the responsibilities that you ladies will be having here today. So Emma, let's start with you. What are you going to be doing?
So I'm going to be actually conducting the post-mortem, so I'll be doing an external examination and then I'll be cutting inside, taking out the organs, giving them a quick check over, and then I'll hand them over to Laura here, who will give them a more thorough check over, weigh them, bag them and record any notes in the Finder post-mortem sheet.
Fantastic. Am I going to be okay if I stand here? Because I'm really keen to sort of get in and
do.
have a look, so it'll be brilliant.
Yeah.
Laura, what about you? What are you going to be doing today?
So today I'm going to be having a little look at the organs to really see if there's any problems or if they're all intact and appear normal, and I'll kind of maybe run you through, like, you know, if they are normal, and if not, what we do kind of look for as well.
It all sounds very, very complicated and all that. Who's paying for this? How is this being undertaken? Are there sponsors?
Yep, so the reason that we're able to conduct this and stream it to people at home is due to the IOSF, the International Otter Survival Fund. So they provide...
Saw the logo here,
yeah,
actually, yes, on the back there, yeah.
The whole day today is World Otter Day, which is kind of the reason that they provided 3 grants. One was luckily enough given to us to do this. The other 2 I think were given to communities in Montenegro and Uganda.
Wow,
yeah,
it's
so
a real worldwide event then,
yeah,
so Cardiff can be quite proud then that we're part of this. This is brilliant, okay.
So they're doing otter surveys to kind of tell people about an otter's habitat, but we're obviously kind of doing a different viewing of an otter from the inside out, really.
Wow. Well, look, I'm very conscious. I'm chatting away here. I know we've got to get underway with the actual autopsy. So, am I going to be disturbing you if I'm chatting to you whilst you're?
That's fine. Please ask questions.
Yeah.
Cutting away. You're sure? Yeah.
Yeah.
Okay. Shall we start to get underway?
Yeah,
and we'll
let's
ask
go.
questions as we go. So there we go.
Yeah.
All right. This is very, very good.
So straight away, I can tell that this is a male otter because there's only one hole, the anus hole here. You can see as well the lump here, which is actually the testes. And because it's kind of a prominent lump, you'd say that the testes were prominent in the scrotum. The next thing I will look for is a general condition, so an overview, so any open wounds, cuts.
And I've got to say, this looks an absolutely huge otter. So it's a male otter, obviously.
Yeah, it's quite a large one.
It's a
for
big
male.
one, isn't it?
Yeah.
Do we know, for instance, how much that would weigh? Do we take that information?
Yeah, so we've got this one down as 8.76 kilograms. So this is quite a heavy otter.
Absolutely. Well, you know, I've only seen otters like this when, you know, you go down to a riverbank and all of a sudden you hear a sort of splosh and then you see an otter swimming away. But it's very, very rare, Liz, that you get to see an otter this close up. And it's just fascinating. I mean, look at that. You've just moved the head towards me. I'm looking at the teeth. They're formidable.
So having done the check, there are no external wounds, so it all pretty appears normal. The next thing I'll check for is the teeth, so I'll see if they're all present. There are 9 teeth on each side, upper and lower, and these all appear present. So
Now, when you say they appear present, what are you getting? If they're not present, what could that mean to you?
So often, if an otter's been hit by a car, it will have a few teeth knocked out, and we'd check if that was fresh damage, or if it's older damage, it could be they could be worn down if it's chewing on fish bones, for example. It's really gnawing away. The other thing we'll check for are chips. So I don't know if you can see here, this upper right canine is.
Oh, I can see that. Yeah, there's a little chip on the end of the canine there.
And is that worn down or is that smooth?
Actually, looking at that, it's quite smooth. So it's probably been worn down, so it's old damage.
So can I ask a really silly question then straight away? I'm looking at those teeth there, and I know that obviously juvenile animals, there will be teeth that haven't migrated, and we can tell if it's a juvenile animal or an adult. But when you've got an adult like that and you're looking at those teeth, is there any way to sort of age the animal from the teeth itself?
Yeah, we actually take the whole skull if possible, and from that we'd extract a canine tooth, and we can send that away for what's called cementum analysis. So very much like trees have growth rings, so do the teeth of an otter, and so by sectioning those teeth and counting growth rings in the cementum, we can gauge how old that individual is.
That is amazing. Emma, what on earth is that?
I've just taken the tongue out. This is one of the first.
Casually, you've just taken the tongue out. You didn't tell us about that.
It does come out quite easily.
So it's not a case of, did you cut that or did you?
Yeah, so we just cut through it with a scalpel.
Okay.
So I'll grip one end and just.
So I'm going to ask the obvious question now. You've cut the tongue out. Is that to aid you to look around the mouth more effectively, or is this tissue of value to you?
So right now I'm actually going to be taking this tissue as a sample. So we just put it in like a little Ziploc bag, and the number of the otter is put on the bag, and also what it is. So obviously tongue for that one. And then we put them all into an archive, and there's like everything is kind of kept there since the very start of the project. And we've got loads of organs down there.
So I mean, from what you're saying then, for me, this is really remarkable, because what you're saying is there's going to be aspects of this otter that you're going to be looking at now for current research. But I take it the tissue like this that you may not be using at the moment, there's more tissue coming, this is fantastic. This is coming my way now. So there's tissue that you're not actually using in research at the moment, but which you're looking at saying, well, this could be interesting in the future. You're archiving all of this.
Yeah, so we've got a big bank of lots and lots of different sample types, which is really important because these sort of long-term data sets and sample banks are really quite rare in ecology, and it forms a really valuable resource that we can share with other researchers, or as things change, often new questions are asked, methods have developed over the 25 years of the project, and the sorts of things that we're able to do have really changed. And also money is a real limiting factor. So as money becomes available, we can say, right, well, we've got this 25-year time series of this type of sample. We can do analyses on those for all sorts of different.
So absolutely fine. And of course, as technology's advanced, I'm just thinking of DNA technology already, even over the last 10 years. So you've got all of those tissue catalogued, archived away. This is remarkable. It's fascinating. Now, I saw something come this way which looked like, I'm going to say whiskers. I bet there's a proper name for it with otters. What's the proper name for whiskers on an otter?
So we've got I've actually packaged them up now. So this is how we normally package them, and they're normally called vibrissae. So that's like the scientific name.
Okay.
So that's them just there.
And what interest would they be, Liz? Why would I be interested in vibrissaes?
So there's a couple of things that we can potentially do with vibrissae, and so one is contaminant analysis. We can look at what contaminants the otter might have consumed with its diet that have been deposited in hair and in vibrissae. So
things like heavy metals or pesticides, maybe. Yeah.
Okay. Similarly, stable isotope analyses. So that's something that we can use to indicate changes in diet. And because the vibrissae are constantly growing, it's a really nice sample type because we can say, well, whatever is deposited in that whisker at the tip was longer ago, whatever's right near the base is very recent. So we can section different points along the whisker.
So temporal differences, time series, that's incredible.
Yeah.
I'm just looking at I'm looking at Emma because she's beavering, well, she's not beavering, she's ottering away there. And I can see that you've been pulling the legs, sort of pulling to see the movement of the legs. What are you doing there?
Well, I was just checking the feet, which all appear pretty normal. We look for things like wounds, lacerations, puncture wounds. So that's why I'm lifting them up. But I am going to be a bit rougher with the bones to check if there are any bones broken, and I'll also check
I
the skull and
see.
spine.
So what, by moving the sort of limbs quite aggressively, you're able to, you probably feel the grinding if there was a broken
any
bone
cracks or fractures, we generally do
Gotcha.
feel them. If we do miss it later on, we can expose the leg bones, so we are often able to see anything we've missed if something is broken. But all of these limbs are intact.
Brilliant.
So we haven't said, Reese, but very often the animals that come in, about 85% of them are actually roadkill.
I did actually wonder this. I was going to say, how on earth have we got this animal here today? So this wasn't killed specially for the autopsy
No,
or something. It's the
no.
European protected species,
Absolutely.
of course.
Yeah.
So roadkill is where all of them come in, or
Not all of them, but the vast majority. So it's about 85% are killed on the roads of the animals that we receive, and so very often that's associated with traumatic injuries like broken legs, spine, skull, and what have you, which is what Emma's looking for. Obviously, if they're killed on the road, they're easier for people to spot and report, so we probably have a bias in
Yeah.
that direction.
And it's unusual for me because I'm sure there's going to be lots of people at home that are looking at an otter and say, well, this is an otter, they live in the water, they live in waterways. Why would I find an otter on a road?
So if you just really have to look at a map of the UK and you can see the density of our road networks and you can see the density of our river networks. We have a lot of water. And what people might not realise is that otters have quite a large home range. So they might be ranging, one individual otter like this might have a range of tens of kilometres.
Wow.
So if it's ranging that far along a river, it is absolutely inevitable that it will have to cross probably multiple roads within that range. And that's where these accidents occur.
And of course these animals have evolved to occupy ecological niches along waterways. They haven't evolved quick enough to deal with our motor cars which have come in, and they come into conflict with them and obviously come off worse. A big male like this, why particularly a big male would be crossing lots of road systems?
So males do have a larger home range than females, and it's likely that a more dominant male, potentially associated with size, would have a larger range. So we do have a bias in that we get more males. More males come into us than females, and that's probably because they have a larger range.
And is it just the adult animals? Is it just the adult males, the adult females, or do you ever get any of the pups in as well?
Yeah, we do. We mostly get sub-adults, and again that's probably biased because we're seeing mostly roadkill. Sub-adults are the group that are dispersing, so they're likely to encounter roads, and they're also possibly a bit less road savvy than the adults, whereas the little juveniles, they're not travelling around as much, so we're less likely to get those also. So it's the sub-adults that are mostly killed on the roads.
Very interesting. Okay, now there's something very interesting going on here because there's a magic machine, a machine that goes ping literally, being sort of sent all over this otter, and it just made a beeping sound then I just heard.
Yeah.
What does this mean?
This is a microchip detector. We run it over every otter that comes in, and it will pick up any microchips if the otter's been released from a wildlife trust or a vet or something like that. It will pick it up. This one has no microchip.
Yeah.
Okay.
And that I think is the last of our external examinations. I don't know if you saw earlier, I was checking between the shoulders and the ears for things like ticks.
So you're looking for external parasites as well as just wounds. So what would you do with the tick? What interest would that be? The ticks, for instance.
So sometimes it can determine how long they've kind of been left at the place that they've been found. So sometimes we even have otters with maggots around any wounds and things like that. So it can often determine the state of decomposition as well. Sometimes we get kind of an idea before we cut in what we're going to kind of see.
Yeah,
What ticks is it? Is it like deer ticks or is it?
So it's actually mostly hedgehog ticks that we find on otters.
Right,
almost
okay.
exclusively hedgehog ticks.
Wow.
So that's quite interesting in itself. Most people are more aware of sheep ticks, and there are also dog ticks. We very occasionally find those, but mostly it's hedgehog ticks. We're also interested in things like tick-borne disease.
Of course, yeah.
So that's something that we can look at by testing the blood and things like that. And also, as Laura says, we're looking partly for ticks, but we're also looking, as well as the parasites, we're looking for any other insects or anything that might be coming onto the body.
So you guys are not going to tell me, you don't catalogue and archive those as well, do you, the ticks?
Yeah, we
Do you
do.
That's amazing, it's absolutely amazing.
We put them in little tubes that look just like this,
Right, okay.
with their number on as well, and we just put them in ethanol to preserve the ticks or any kind of ectoparasite that we do find.
I'm just thinking, what a resource this is. I'm just thinking, I come in 10 years down the line wanting to do a PhD in otter, ticks that are found on otters, and the archive's all there. It's just all there instead of trying to collect all of that data.
Yeah.
It's absolutely incredible. So when we're saying we're archiving this from various animals, what are we looking at? 2, 3, 10, 100? How many of these otters are you getting in a year from across the UK?
We get around 250 a year.
Wow.
From the start, we've actually numbered them, and this is the number we have. So 3,313. This is
That is how many you have in the archive at the moment.
Yeah. Since the project began.
I don't know if I'm horrified at that, to think and that's primarily roadkill, the conflict of cars and otters. It's horrific, isn't it?
It's important to say though, I think of course it's really sad that these animals are getting killed on the roads, but the increase in the numbers that we're seeing killed on the roads actually is in part reflecting the increase in populations. So yes, partly it will have increased because our road traffic has increased. It will also have increased a bit because awareness of the project has increased. But probably the main driver in the increase is actually recovery of the otter populations themselves.
Do you know what? I had it in my head, and I'm going to be corrected now, ladies, I know, but about, back in the 50s and 60s, I was thinking that otters were almost occupying a small niche down in sort of mid-Wales. And then outside of that, there were very few large populations of otters.
Yeah.
Is that correct?
There were kind of several areas where there are remnant populations. So the southwest of England, some of the southwest corners of Wales, Scotland, and a little pocket in East Anglia. But yes, those were kind of remnant populations. But they've really expanded back from there, and they've now been seen in every county in England again, which is amazing.
So the question has to be why? Why are they expanding?
We've cleaned up the habitat.
Right.
Chemical contaminants, which is one of the things we're looking at in post-mortem. Those chemical contaminants that were harming populations have decreased and populations have recovered.
And our river systems as well cleaner? Is that going to influence the otter populations? I've got to say, the thing that's surprised me more than anything today is the fact that at the moment there's very little smell. I expected it to utterly stink in here.
It's get a lot worse.
Is it?
Like,
Oh, right, okay. I saw the scalpel coming up and I thought, let's see that. And of course, what we should also just warn you guys looking at home now, it's a real autopsy, so I take it that there's going to be quite a bit of blood and...
I can see here there's a little bit of kind of loose, mushy feeling stuff, so there might be quite a bit of free blood in the abdomen, but again, we'll find out when we cut inside.
Okay, but it is going to be quite graphic. We're going to be cutting into an otter here, so just please be aware of that when we do that.
Yeah.
So all the external observations are done?
Yeah, we've just gone through the list to make sure that everything is done, so we're all good to go. Get on
pretty
to internals.
in good condition otter. He doesn't even have any fighting injuries, which I checked for earlier.
When you say fighting injuries, what do you mean by that?
So again, it's things like puncture wounds, lacerations, and they'll be around the anogenital region, which is just...
from male otters fighting with one another?
Yes.
They target that area. Yes.
Don't mess with an otter.
Reproductive consequences.
Don't mess with an otter. Goodness me. Okay. Yeah. Well, I mean, even in my unqualified eye, you can see that's looking relatively clean. I'm just still in amazement at the size of this animal, and it's so beautiful. I mean, when you see these on a river, it's interesting. But to see one up close here, I'm looking at the feet, and I can see the little webs in between those feet.
Got beautiful markings also, that one.
Yeah, so we've made a note.
Do you know what? That looks unusual to me. Is that unusual to have markings like that?
It's very variable. Some of them have a completely brown throat, some of them have like a single white spot, some of them have lots of spots. It's really quite variable.
Is it a way to ID individuals?
It could be, if you could capture that part of the throat reliably on camera.
Absolutely.
But when they're moving around at night, that's also very tricky. So.
Absolutely.
This one also has a slight white moustache here.
He does, doesn't he?
And then a generally quite pale underside and bib area, which is just this area around the throat, chest.
What an incredible animal, though. A native British otter is a quite incredible animal.
It's about to get not quite so incredible.
We're about to see what's on the inside now.
Yeah.
So this one is I think from the weight that Laura suggested earlier, it was 8 point.
So 8.76.
Yeah, that's just a little bit above average for an adult male. Adult males average about 8 kilos. Adult females average about 6 kilos. So he's on the big side, but we've had bigger. I think we've had up to about 12 plus kilos.
Wow,
Which
that's
The one the other month was 12.
that's a monster. Did he fit on the table?
yeah.
Just about.
Yeah.
So what are you doing now? What's the start of the autopsy here?
So I'm just running the scalpel quite lightly along the pelt to peel back the pelt layer, which I'll do in a second. So I'm just going down to the fat layer. I'm not going straight into the abdomen, which is why the smell won't come out straight away.
Okay. I've got that joy to come by the sound of it.
It looks fairly fresh, this one though. I think the state that they come in can be very varied,
Yes.
as you can imagine. But this one so far looks pretty good. I think you're in luck.
And the pelt comes away quite easily, doesn't
So
it?
we
It peels
just
away.
pull the pelt up like this, and you can see this little connective tissue here.
Yeah.
We just
Gently.
lightly.
That must be quite easy. That's a skilled hand there, making that look very easy, because I
It
can
is
guess that you
surprisingly
can
easy.
you can cut through that in all the wrong places.
So Emma and Laura, we've been training them this year. So they're both here on their professional training year as part of their undergraduate degrees.
Okay.
So during this year, we've trained them both in this standardised method that we're using to do the post-mortem. So they have both had quite a lot of practice by now.
Yeah.
And they're doing a great job.
Well, what an opportunity! What a fascinating opportunity to be able to do the professional training year at Cardiff University. And how have you felt undertaking that?
Yeah, it's been brilliant. To be honest, before this placement, I've never got up close and personal to an otter, and I never thought I'd be doing this, but it's actually been a really amazing opportunity, and it's been really fun to learn a lot more about otters.
That's amazing. I don't think many people can put undertaken. How many autopsies do you think you've undertaken?
A lot.
A lot.
Yeah.
Well,
Over 100.
Yeah.
over 100 autopsies on your CV. That's not bad to put, is it? That's going to put you ahead of the game.
So you can make a note of this bruising
Yeah.
to the lower, well, actually just left side of the abdomen.
Okay.
So this is quite indicative of possibly being hit by a
An
car
impact,
around.
yeah. So I don't know if you
around
guys can see
area.
this at home, but what we've got is you can see a lot of bruising, and there's a lot of a pooling of blood, shall we say, on that side,
Yeah.
which could be indicative of a
Heavy
car
impact.
glancing it, maybe.
Yeah.
Yeah.
That's not going to do it any good at all, is it? That's
You can also see sort of across the surface of the body, you can see the fat layer. So that paler material is the fat.
sure.
Yeah. So this.
Yeah. So I think very often people, they think kind of aquatic mammal, and you're expecting this thick blubber
Lots and
layer,
lots of blubber, isn't it? Yeah,
like a seal or
Yeah,
something.
yeah,
but
exactly.
that's not the case. They have quite a thin fat layer
Right.
right across the surface of the body, with some areas where you tend to get a bit more around the abdomen.
What is? I can see exactly what
So
that is
this-
now. Yeah.
This is the penis bone,
Right.
also called the baculum.
Baculum. I was going to say that. I knew that. Look at that. There we go.
So I'm just going to trim
Wow.
the excess tissue off here, and we'll then measure the length of it, which is how we kind of do the age classification for males.
Through
So-
the baculum. Is the baculum that classifies the age?
Yeah.
That's remarkable. I
Yeah.
didn't know that. Because we're quite unusual as a species, aren't we, human beings? Because we don't have a bacula, of course.
Yeah.
We're I think most mammals do.
Most mammals
Yeah.
do, yeah.
Yeah.
Yeah, and so we measure the length, and it's one of the age indicators that we're using. Obviously, we can't use it for females.
Yeah.
But yeah, for a male, we would expect that an adult male would have a baculum of at least 60 millimeters long. So we'll check
Wow.
the length of it, and also
That's
weigh it.
quite incredible, isn't it?
Yeah.
Look at that.
Give that to Laura.
Thank you.
Sure.
And so, there must be other information you can get from this as well.
So we look at the baculum. One of the things that we're looking at, we'll kind of feel along the length of that. We'll check it in a minute. We'll see. Is there any indication that it's been broken? That's something. We talked about those aggressive
That made me wince
interactions.
then. That made me wince, yes.
we do occasionally get individuals that have had a completely broken baculum, and then we can see where it's rehealed and a callus is
Right.
formed on the bone.
Right.
So that's a very severe end of those
Yes.
aggressive interactions. We've also done some research in the past looking at the density of the baculum.
The
so
density of the bone
density.
itself, okay,
Yeah.
and what would that indicate?
So it might be that changes in bone density might relate to things like aquatic contamination with chemical contaminants. But in this relatively small study that we did, we didn't find that association.
Right, okay. This is utterly fascinating stuff. I'm kind of right in there, like she's in there.
Yeah, so
So-
I'm just peeling back the next layer.
Okay,
Just
and
the
what is
muscle
that next layer there?
The muscle layer.
Okay, yeah.
So I don't know if you can see the thickness there. We kind of put 2 fingers across and make
So the
kind of a
pecs then,
judgment
I guess.
call
Okay.
of how thick the layer is. So I would say this is a pretty good muscle layer. We
I mean-
go from poor to moderate to good, very good, extremely good. But do you think that's good?
Yeah, I would say that looks good.
Yeah.
Because it look, to me, Liz, that's looking like a healthy animal on the
Yeah,
inside, yeah?
Yeah, it's a relatively subjective measure when
Of
we're
course.
looking at those broad categorisations of is the fat layer normal or is the muscle layer normal, and as much as anything it's picking up if there are those kind of really unusual cases where we're seeing emaciation and
Yeah, cool.
muscle wastage and so on.
So,
I was going to say, Laura, what are you looking out for at the moment? Because I can see you hovering there and you're grabbing pieces of tissue as you go.
I just
what are you looking for?
thought before I put the baculum away to just give you a little bit of a
Sure, yeah.
look at what's happening. So I can tell that there isn't any previous injuries. I don't think there was a breakage here before, and it was just 71 millimeters as well. So we can say that that is an adult.
Great.
Yeah. So then again, that gets put in a bag then to be put into the archive.
Okay, fabulous.
We work with National Museum of Scotland for archiving of the bony material, so they take the bacular skulls and leg bones, so they have an extensive archive now of these.
It just goes to show the importance of this research, because I mean this is UK-wide, isn't it? It's not something that we're just undertaking
Yeah.
here at Cardiff.
Yeah, absolutely.
It is quite an incredible operation.
Supported by a huge number of organisations, both in terms of collecting the carcasses, reporting the carcasses, helping with archiving, collaborators on the research and so on, yeah.
I just thought as we got to this point as well, we should point out that it's not just us here today. While you're watching this at home, we've got a team of young ladies upstairs. I'm going to give them a shout out. Nia, Juliet, Laura and Amy are upstairs on the computers, and they're going to be answering any of your Otter questions. You can get hold of us over social media or via email. Email, it's going to be otters@cardiff.ac.uk. We've got on Facebook, we've got the Otter Project at the Otter Project UK, and we've also got on Twitter there, you've got Ask Otters 2018, and all of that information should be coming up on your screen now. Please do contact us, ask us your Otter questions. I'm trying my best here to anticipate what you want to know, but there's probably going to be specific questions I haven't asked. Ask the girls, they're all eager to answer your questions. Where are we?
So I've just trimmed the muscle layer so you can see the rib cage here. We're going to go into that later once we've done the abdomen.
Okay.
So this is the final warning that the smell is about to get a lot worse as I go in.
Okay. So I should notice a difference now in straight away. Here we go.
So this is just the muscle coming off.
Still not too bad.
Oh, it's
I'd
not
say.
too bad, is it?
No,
I thought it was going to be
it's not
lots
too bad.
worse than that.
Yeah.
I was really prepared today for something that was going to be really
You
smelling.
can normally tell the bad ones as soon as you open the bag, so.
I guess the older or more sort of decayed the animal that you get in, it's going to smell worse. But
Yeah, that's
I
right.
have to say,
Yeah.
I think you've been very nice to me today. This
Yeah,
looks
I think
a
so.
fairly fresh animal.
Pretty good.
Yeah,
So then,
often
yeah.
sometimes we have, sometimes because of the trauma of the RTAs, we have stomachs that have been ruptured, and that's when the real smell happens.
Oh.
So we'll make a quick abdominal overview. So this all appears to be slightly pulverized, probably from the impact where we saw the bruising earlier, so around this area.
It's quite traumatic, isn't it? That?
We'll check if this kind of skin layer over the top called the peritoneum is intact.
Yeah.
This appears actually to be intact, although it is starting to disintegrate.
Yeah.
Yeah. And the diaphragm there, if we can check that.
So the diaphragm is there. It's helping the lungs. It's pushing
So it would
the
normally
animal to breathe.
It is actually intact
Amazing.
here.
Great.
Yeah.
So that separates the thoracic contents from the abdominal contents. And very often, if there's been an impact on either area, it ruptures right through, and then you can get thoracic organs. So the heart, for example, might come down into the abdomen, or parts of the lungs sometimes we find in the abdomen, and vice versa. We'll get abdominal organs pushed up into the thorax.
So if I pull up the peritoneum here, you can see the spleen starting to emerge.
Oh yeah.
So it's quite bruised.
So that sits on top of the liver, doesn't it? Is that right?
Just sort of to the side of it. Yeah.
So I'm going to cut this out so you'll be able to see it a bit more clearly.
There we go.
It's also slightly fragmented.
Yeah.
So we'll make some notes.
So what is that telling you?
Really just about the impact. We do keep the tissues. Again, it's something that we're keeping for the archive. At the moment, we're not doing any specific research using the spleen, but it's archived as a just in case.
Wonderful.
And we would also, all of these organs, we would weigh them. So Laura would be weighing, if it was intact, Laura would be weighing it. There's no point if it's fragmented because it's not representative. But yeah, we're kind of looking at the weight of the organs relative to the weight of the otter. So that we can see,
makes sense.
Is it undersized, oversized, does that tell us something about the health, and so on.
Yeah, so we can see with this one, as Liz and Emma both said, that we have got a bit of fragmentation just there.
I can see that. So is that literally ruptured, that's ripped,
That's
due
it,
to
yeah,
impact?
from the impact.
Wow.
And it is a little bit darker than normal, which is probably due to bruising, again, from that impact. So normally we would cut that in half and keep the sample, but for this one, because it is fragmented, we won't take the weight, but we will keep a sample.
Okay.
While Laura does that, I'll just say I've just taken out the bladder, which sits around the lower abdomen, as you'd expect. So it all appears intact and it's actually empty. So we'll just make a note of that.
Now when you say empty, are you talking about urea, or are you looking for anything else in the bladder?
So we check if it's full, obviously, of urine, or whether it's bloody urine, whether it's just blood. We do occasionally find very, very small pieces of sediment in the urine, which we'll make a note of as well.
And very occasionally stones we end up with in the bladder. If there's been a kidney stone that's shifted downwards, sometimes we might find that in the bladder.
Because I get, of course, because of the diet on these guys, they have kidney stones often, or
fairly often. Yeah, we find kidney stones in about just over 10% of the adult otters. And yeah, probably relates to the very kind of high protein
diet. Yeah, that's what I was thinking.
So that's something, when Emma gets onto the kidneys, that's something we'll be looking for.
Okay.
So I've just clamped up kind of the top end of the intestinal system and the lower end. This we'll be collecting fecal samples from, which is why Laura's just put the potato on the table, and we'll then empty the stomach.
Okay.
So as I've clamped it shouldn't come out straight away, but I'll just cut.
So I mean, is the stomach of interest to you? Are you able to ascertain anything about the diet of the animal from the stomach contents? Yeah.
Yeah. So we actually retain the stomach and its intestines, and also we have a look in the stomach as well, and we take its stomach contents, and we also kind of have a little fish around to find some fish, or maybe different kind of things that it's been eating.
So what unusual things have you found in the stomach before then? What's give me some ideas of some of the things you may find in an otter's stomach.
So one of our most interesting ones was when me and Emma first started, and we actually found some octopus tentacles.
Seriously?
Yeah, so that was an otter that was found in Scotland. So because they are found in more coastal kind of ecosystems.
That's incredible. I never would have thought that. But
most of the diet is fish based. And for that we'll be collecting any of the remains that we can. We clean those up. We can look for hard parts like the vertebrae, which we can identify at least to family level, and in some cases to species level.
Identify the different fish with otters. That's incredible.
Yeah. They also eat, they're very opportunistic, so at certain times of year, for example, when the amphibians are coming into breeding migration and there's concentrations of amphibians at the breeding pools, we'll find a lot of those. They will feed on crustaceans, particularly things like crayfish. Also the coastal otters will be feeding on crabs.
Have you ever found anything like, I don't know, bird feathers or anything like that?
Yeah,
So they
just
will
a few
even take
days ago.
birds.
Yeah.
Really? Wow. Okay.
So
I love this. Look at this now. Let's go in for it. You're emptying the wow, the intestines and what have we got?
Can be
in
a
there?
bit tricky because they're a bit slippy and there's not a great amount in here.
Slippy intestines.
That's it, yeah.
All right. Here we go.
So there we go. We've got our 2 fecal samples just there.
Wow. I've got to say this is such a fresh otter because
Yeah.
it's still not an overwhelming
Yeah.
stench yet.
Yeah.
It's pretty good.
So the next thing I'll do is just empty the stomachs into one of these tubs.
That's the part that could smell a little bit.
Okay. Here we go. Pocket of digestive juices, really, isn't it? So if anything's going to smell, this is going to be it.
Yeah.
Oh, you can certainly. It's gone up a notch, hasn't it? That's for sure.
So
Not
I'll
too
just
bad, though.
turn the stomach completely inside out. You can see there the kind of ridges on the inside of the stomach lining.
Yeah, right.
So we'd also be able to see if there was any kind of ulceration or anything like that in the
Of
stomach.
course,
on the
yeah.
inside by there.
Okay, Liz, what we got in there? What's that?
So it looks like we've got a bit of fish there.
Fish.
Oh, I can see that's the
fin
fish
there,
tail. That's
yeah.
Yeah.
Wow.
You can kind of see, you know, like the texture, you know, what it actually
Yeah.
looks like. You can tell that there are some scales there.
Yeah.
Right. Okay. Look at
So
that.
yeah, I wouldn't just by looking at it be able to identify the species of fish from that. The best way to identify the species on those hard parts is by looking at the vertebrae, and there are keys that have been developed so that we can look at the spiny projections
Wow.
on the vertebrae to identify. But
Amazing.
the other method that we can apply to these samples, and one of our PhD students, Lorna, who is there ready to
Yeah,
answer
she's
questions.
upstairs typing away. Yes. Yeah.
So her PhD is looking at specifically at the otter diet, using both kind of hard parts analysis like this, the traditional analysis, but also more recent techniques like using the DNA that we can extract from the samples.
I was going to ask about that. If you could use DNA to actually determine
Yeah.
exactly what you're looking at, screen for that. So could
Yeah.
you screen for lots of different sort of prey items and in one sort of
Absolutely.
go?
So she's developed what we call primers, which are it's a way of kind of fishing for specific
small little
DNA.
sequences of DNA, aren't they? The
That's
fish
right.
for bigger sequences. Yeah.
And we can target then primers that will pick up any fish, and then we can look at those sequences and identify those fish to species, which is much better in terms of the resolution that we get than the hard parts analysis, where for some of them, for example, the cyprinid family,
Yes.
which is a very diverse family of fish,
Yeah.
the vertebrae all look the same. So whether it's a roach or a rudd, for example, we can't tell
So
the species from the hard parts.
morphologically, they're just almost identical. You
In
can't
terms
tell
of
them
those
anything, are you?
vertebrae, which is what we
Yeah.
mostly pick up, yeah.
Right, okay.
Occasionally, if you get a jawbone in the sample, you can identify to species, but you have to be quite lucky to get the right part of the fish, and most of what we get is the vertebrae. So by using the DNA, we can get a much better resolution. We can get right down to species level.
Absolutely fabulous.
And it also really helps if we've got things sort of in the gut. We might not necessarily pick up hard parts. So
Yes, no, absolutely, yeah.
some prey don't
Yeah.
have hard parts. In some cases, the otters might be, if they're taking a large fish, for example, they might strip the flesh away,
Yeah.
but not eat the spine.
Yeah.
So we'd not detect it using those traditional
Yeah, or
methods.
certain invertebrates or something like that,
Yeah.
they may be very
Yeah.
subtle. You're taking blood samples now, I noticed,
Yeah.
Laura. So what are you doing with this then?
So we put those in these little Eppendorf tubes, and again we retain those and we keep the 3 samples. So we do normally try and take it from the thoracic cavity or the abdominal cavity as long as the stomach hasn't been ruptured, because then it can be contaminated with any of the stomach contents. So yeah, we've got our 3 samples ready to go
Fantastic.
there.
I'm going to ask why you were taking blood samples, though. I can see we are. Why are we taking them, Liz?
So that's screening for disease is something that we can do. So we
From
can
the blood sample?
screen for antibodies against disease. So for example,
of course.
we've done some research looking at the presence of antibodies for Toxoplasma gondii, which is a parasitic infection.
Yes.
And we've been able to determine that by looking at the seroprevalence, so the prevalence in the blood.
Utterly fascinating. It's amazing. Just we've only just started the autopsy, and the amount of information we're able to sort of yield from this sample already. And we're only in the abdomen at the moment. Is that a kidney?
Yeah, so I've just taken out one of the adrenals, the right adrenal.
Okay.
I
Just
can't.
here.
Oh, that there. I'm
Yeah,
sorry, I'm
that
looking
little
over the top
bit.
there.
Yeah, that little thing there.
So the adrenal glands, to me, that's sort of you know, basically when I'm stressed, when there's adrenaline.
That's it.
That's the adrenal gland. It's absolutely minute. I've no idea how you found that with all of that
I
mess
actually
there.
can't find the left one.
Oh, there we go. I feel better now. I feel better. I was going to say that was I would have been lost there. But that's absolutely, well, I say it's absolutely minute, ladies. Is that the right size? Is there anything you can tell me about it?
Yeah, so we have a look at this, we check if there's any kind of nodules on there or anything like that, and for this one it appears intact, and it also appears normal as well. So this is a normal kind of looking adrenal.
And is it sort of about the right size you'd expect for an otter of this size? Because I suspect it's relative, isn't it?
I mean, directly from the post-mortem we couldn't say for sure. It looks broadly normal, but we would check the data afterwards. So Laura will weigh each of the adrenal glands separately, and again we'll look at the weight relative to the weight of the otter and the length of the otter, and we'll say, you know, is that normal or not?
And if it was enlarged, could that be that, I don't know, the animal was stressed? Or
so any form of physiological stress, and that might be ill health, but it might equally be something like pregnancy. So pregnant females often have enlarged adrenals.
Yeah. It's a stressful time, this.
Absolutely.
It's a stressful time.
Yeah. So, I mean, in terms of, you mentioned, and Emma said you couldn't actually find one of the adrenals. This is the difficulty when you're trying to do dissection work on something that's been hit by a car. Things aren't necessarily where you expect to find them.
Exactly.
And a lot of what we're doing, if the kidneys were in the normal place, we'd be looking for the adrenals at the top of the kidneys. But if those kidneys are detached, disrupted, or whatever, it can be very difficult to find.
Now, I just want to mention something I've noticed, because this is really interesting. Because when I think of kidneys, when I'm looking at a human kidney, for instance, it's very, very smooth. It's like a lobe. And this is, what would you call it? Is it reticulated, or is it,
So you can see that it has many lobes, unlike the human kidney. And we actually look into each of these lobes to look for any kidney stones. So kidney stones you can kind of see from the back of the kidney. They normally start just inside in all the tubules, and we have to check all those.
Are they visible with the naked eye, the kidney stones? Or
very occasionally, actually. If it's a really severe case, and so normally when it starts, it's in the middle of each of those lobes. So Laura's just
so you have to dissect into lobe, right?
And
okay.
In the middle of those lobes, you can see where the ducts
kind of
are
just seen here.
Emptying out.
I can. There's some little tiny ducts there connecting each of those lobes.
And if kidney stones start, it tends to be just a tiny little granule in the middle of that. But then as they grow, the sort of concretions increase and increase on that. You still can't see that until it gets quite large. But sometimes when they're very large and they really disrupt the function of the kidney, and they grow into what we call stag's horn calculi, where they're kind of growing out through the tubules, merging together between lobes, and then you get to a point where you can see that externally, because the lobes of the kidney are dead, not functional, and you can feel that the whole thing really can turn into what feels like a rock.
So is that age related? Is it diet related? Is it both? It's both.
Both. Okay. So they will tend to accumulate with age, so we find they're more likely in older otters, but it's also diet related, down to the high protein content of the diet.
And you can see that this left kidney is a bit more damaged, and if I put them side by side, you can see that one is slightly darker as well. So again, because of the bruising due to the impact of the car. So again, you can have kind of like, you can see a little bit more into this one, but I still will cut it open just to make doubly sure that there is no kidney stones in there.
Wow, so it's just...
make a note that it was partially detached as well in the abdomen, which is probably why the adrenal is lost somewhere.
It's in there somewhere.
Wow. Sometimes we find these things later, and you learn to recognise tissues by feel and by texture. But yeah, sometimes
it's going to be very difficult to appreciate just how tiny the adrenals were, but I can tell you from someone over the time, I mean, it was basically the size of my little fingernail. Utterly incredible. It was small. Just while I've got you there, ladies, I'm just going to remind the people at home, don't forget you can ask your questions. I'm asking everything that I think you'll want to know, but of course you yourself can ask on Twitter, #AskOtter2018. We've also got on Facebook at Otter Project UK, and of course email as well, otters@cardiff.ac.uk. Get your questions in. We've got the ladies upstairs. Keep them busy. They get bored otherwise. Ask your questions, okay? And I'll try and anticipate what it is you'd like to ask. Do my best, but remember, you can ask your own questions too. Okay, so where are we?
We now have a quite severely autolised liver. So
Wow.
usually the liver is a pretty smooth texture, as you can see here, but it's
So it's very fragmented,
Yeah.
kind of almost pulverized. I'd say it's probably not autolysed as such. Autolysis is when it starts to basically break down and decompose. And as I said, I think this one's fairly fresh. It's just that impact has really smashed it to pieces.
Because the liver basically is there, it's helping to filter the system, isn't it? It's helping to get rid of toxins, just in day-to-day diet as well. So, I mean, that to me is looking as if it's had a fair old smash and it's been ripped apart.
Yeah.
How would that affect an otter? Would it kill the otter? Or, that would kill the otter. I mean, to me it's looking like it would kill the otter, but I don't know how hardy these otters are. I think you'd have to be pretty hardy, wouldn't you? So that would be fatal, and you can see that. The more you spread it out, you can see that it's just.
So the level of internal bleeding there is huge.
Yeah, you can see there's a lot of free blood.
Oh, and now you can really see. So it's pooling in the side, and the pelt is now pooling blood there. So
I'm just going to take a pelt sample actually before it gets too kind of wet with blood. So I'll just cut that off and lower it back.
So, I mean, you've just said there that you're cutting a pelt section. What information can we get from say, the pelt as well?
So again, as with the liver actually, it's taking them at a similar time. The contaminants are a really interesting focus of the project because the otter is top of the freshwater food chain.
Like an apex predator, so
so chemical contaminants that end up in our waterways, things like pesticides, flame retardants, all sorts of chemicals, they end up in the water, but they can be very, very, very dilute, so very difficult to detect in water. But by the time they accumulate up trophic levels, so small fish to bigger fish, and then finally to otters, each level they can accumulate, and so we can detect some of these chemicals in otter tissues that we wouldn't necessarily detect in water. And because the liver is responsible for that cleanup of chemicals from the blood, that's an organ that we tend to target for chemical contaminants. But we can also use hair for chemical analysis as well and see what's being deposited in the hair.
Yeah, because I know in some forensic studies you can look for traces of arsenic or whatnot in human hair, in certain murder scenes and whatnot. So it's the same here for otters. We're able to look, those contaminants will build up in the pelt. Wow.
Yeah, and so also the difference there, I mean, we know if it's turning up in the liver, we know it's become systemic. Whereas if it's in the hair, it may have come from sort of the internal pathway, but it may also be deposited externally. So it's quite interesting to compare those things.
Interesting.
And also with the liver, I didn't say it's very different to the human liver. It's kind of more like it's nearly like a flower petals kind of thing.
Is it right?
Yeah.
Structure.
So obviously you can't really tell here, but you
it's
kind
a squash flower.
It really is. So you can kind of tell the separate lobes that you can see here. And we also do look for the gallbladder, which is normally embedded in that petal.
Okay.
But we can't see that, which is probably ruptured.
Yeah,
of course.
Yeah,
we jump out the way. So what would the gallbladder tell us? What would we find out from the gallbladder? What information?
Yeah, I think we've got the gallbladder there. It's
oh, yeah.
Oh, that's hard to see though, isn't it?
Yeah, it's quite tricky. So if it were intact, we would expect to see basically a small balloon filled with fluid. If you can trim that out.
And that's aiding in digestion again, isn't it? So
so in the gallbladder, that's where we find the bile, and the bile is helping with digestion of fats. So if there's a problem with the gallbladder, then we can see individuals can end up starving because they're unable to digest fat. Even no matter what they're eating, if they can't digest it properly, we can see these problems. And what we look for, not normally at post-mortem, but afterwards when we've taken that sample away, we'll go through a batch of them microscopically and check for parasites. So we find trematode worms in the gallbladder sometimes. So that gallbladder has the texture of that one.
So this one seems quite it seems quite like it appears normal. There's no thickening or anything that I can feel, and it is empty as well, which could be also due to the trauma. As you can see, the liver, you know, and it is a kind of more of a thinner tissue.
It's normally, it is very like the texture of a balloon. It's very thin, skinned balloon. But if the parasites are there, we see thickening. It can become shrunken and thickened, and that's our first sign that there may be parasites there. But we wouldn't take that as a definite. And sometimes they can appear quite normal, and then we dissect them and we do find parasites. Sometimes they appear thickened, we dissect them and we find that there's nothing there. So we always check to make sure.
Do you know what, Liz? I've just looked up and Emma's just completely stripped all the flesh off that one. She bit dab-hand, wasn't she, at a barbecue? That's all I can say. That is amazing. Look at that straight away.
So I will be taking the whole leg off, but the first thing I've done is just taken off this big chunk of muscle. Laura is then going to cut that up into some bigger sections and some really, really tiny sections that we'll put in Eppendorf tubes. Some of them with ethanol, some of them without ethanol.
You can see all the beautiful striations.
Yeah, it really does look quite like beef or something you would find in the butcher.
It's quite rich, doesn't it?
Yes.
I'm thinking of tea now. I am. And of course now you're getting down to, you can see those back legs. So where before you were testing to see if there was possibly any breakages in those legs, now you can physically see them, can't you?
Yeah. Sorry about the crunching
noise. There's definitely a break in there now.
So that muscle tissue, again, Nia, who we mentioned, who's in the background answering questions. So her PhD is looking at the genetics of the otters, and the muscle tissue that we're getting from post-mortem, we can use that, we can extract DNA from the muscle tissue, and we can use that to look at relatedness of individuals, and we can look at the overall population structure and how that's changing.
And that's going to be fascinating because I guess the structure has changed so rapidly over such a short period. There's got to be sort of high relatedness in certain areas, and we can sort of identify those areas where there's larger genetic diversity. Very, very interesting.
And what's also really interesting now is what were those kind of separated populations where there'd been this massive decline, and you had these isolated populations in different parts of the country. Those have now recovered, and they're all contiguous, and so presumably mixing. So that pattern in the genetics that we're seeing is one of the really interesting things that will come out of Nia's research.
Right. And interesting to look at that over time as well. You know, while we've got them, looking at that temporarily. Laura, you're butchering away there.
I am.
What are you looking for now? You're just basically taking samples at the moment, rather than looking for
so I was just trimming it a little bit just to get rid of any connective tissue, because we want the sample to be as pure as possible,
of course,
especially when we're dealing with the muscle. So we take 2 bigger chunks that we freeze, and we also take some littler chunks, which you'll see in a moment, which some of them are preserved in ethanol and some not. So
with some tissues we try and take them in multiple different ways, because we never really know. It's hard to anticipate what the researchers of the future might want.
Absolutely.
Or what we might decide we want in the future as these different methods develop or whatever. It's like for some analyses, tissue being stored in ethanol is perfect. For others, we might want tissue that hasn't been stored in ethanol.
Absolutely.
So try and anticipate those things and take stuff in multiple ways if at all possible.
And I guess probably some people at home were wondering why we're having masks on and whatnot today. But of course, when we're doing undertaking genetic research on these samples, of course, it doesn't matter. Our human DNA could literally contaminate those samples. It wouldn't matter for what we're looking for.
Yeah, if we're using targeted primers that are targeted, so in Nia's case where she's trying to capture the otter DNA, then she wouldn't be coming up the human DNA. In other areas, it can, when you're looking at, for example, the dietary research, more general mammalian primers, because we might want to pick up other mammals that the otter might have eaten, might pick up human DNA, but obviously we can exclude that from the analysis. So I think we're pretty confident that they're not eating humans.
We're very hopeful otters are not eating humans. Of course they're not. They're not. And that's brilliant. And of course the zoonotic disease, there wouldn't be transferable diseases from here that would be of any problem for us either, I'm guessing.
There's a very, very slim chance, for example, if an otter had something like TB. So we are careful when we open up the thoracic cavity, and if there were signs of abnormalities there that might be indicative of that, then we would actually halt this now completely. and we'd take suitable precautions.
But I mean, pretty much most mammals in the UK can carry TB, can't they? From the farm
absolutely.
cat to a vole, a mole, an otter, to anything else, a badger, of course, which always gets a bad name. But there's all sorts of animals that can carry TB. So that's very interesting. You'd open up. But what would be the signs that would indicate that an animal may be contaminated, may be carrying TB?
We'd see lesions on the lungs, and we do check all the lungs carefully. Again, we're looking for any sort of abnormalities on the tissue that we can pick up.
Okay, very interesting. So I'm keeping an eye on her. She's steaming ahead here. What is she doing now, Laura?
So-
What's she doing?
So right now you're getting the testes?
Yeah.
Yeah.
So this lump that we mentioned at the start during the external examination, if you remember I said the testes were prominent in the scrotum.
Yeah.
So I'm now almost going to
Turn it inside
turn
out.
it inside out.
Right,
yeah.
okay.
And trim the fat away, and you can see the testes starting to emerge here. And I'll
It's a
just cut.
Fascinating insight.
And this tube next to it, which you'll be able to see more clearly in a second, is called the vas deferens.
Mm-hmm.
That will all come out as well, and then I'll hand it to Laura, who will very nicely trim it.
So this is all kept as well? The testes are kept?
Yeah.
Or
Yeah.
what
So
a
again,
painful tissue.
that sort of trimming which Laura's doing, it's because we're weighing everything, we want to make sure that those weights are as consistent as possible. So we need to always try and make sure that if we're weighing an organ, we're weighing it as a discrete organ, not with lots of associated tissue and so on. So we try and be very consistent in the way that we take things, and standardised in the kind of the length of extra tubule that we take and so on.
It's utterly fascinating.
And also, I was just talking to you about the muscle just a moment
Yes,
ago, about the different samples. So in a moment, I'm going to be cutting this big chunk into half,
Okay.
and that will be wrapped in foil and then put into our archive. And we've got these bigger chunks of, they're smaller compared to that,
yeah.
but
It's all relative.
yeah,
It's all relative.
these are our big chunks that we just put into these little Eppendorf tubes, and they will be again frozen. And these little ones are actually put into ethanol.
Okay, yeah, so it's all different types of storage
Yeah.
medium for those different tissues, and it's very wise. And has this been going, is this how long have you been storing samples in that fashion?
I think our earliest samples are from 1992.
Wow.
So
Okay, so that's literally thousands then of
yeah,
otters still.
it is. Yeah, so it's over 3,000 otters. The project actually started in 1994, but some of the otters that came in, someone had in a freezer a couple of years before that. So yeah, our earliest samples are back to 92. And that length of time is really important, because that sort of long-term archiving is really rare in ecological studies and so on.
So when the Cardiff University Otter Project first started off, you said in 92, was that? What type
94.
Or 94, what type of area were you covering then? Was that the whole of the UK then, or?
Not then, no. So at the time, the area of southern and southwest England was covered by a vet called Vic Simpson, who's a really experienced wildlife pathologist. And I went to him for some training when I first started, because I started in 2004. The project, we've gradually expanded our remit. Vic has actually retired now from this work, and I think that was in 2007. I started covering southern and southwest, and then Scotland didn't used to be included. The work used to be funded by the Environment Agency, and they only cover England and Wales, so that was the area that our funding covered. And then more recently, in 2014, we started covering Scotland as well. So we're now covering the whole of England, Wales and Scotland.
And what sort of, I mean, over that time period, that's a heck of a time period, what would you say is the sort of most significant population difference that you've seen? You know, have you seen an expanse into a certain area that was absent from otters before? What's
so
been most significant?
the sort of area in central England really was, we very rarely got otters from there, and we've gradually seen an infilling there, and also sort of Thames area. we've seen a real increase in the otters that we're getting from there. It's still very few.
I was going to say they sound very industrial areas. Is that anything to do with the fact that you weren't getting otters?
Yeah, I mean, areas where there's a dense human population, they're less suitable for otters, but we're increasingly seeing otters right in the middle of our towns and cities. Here in Cardiff, we've had footage of them.
Cardiff Bay, exactly. They've gone all the way down the Taff, and there was sprint otter poo that we were seeing left all the way down to Cardiff Bay, which I was fascinated with. They've gone right through the city. So it just goes to show how green Cardiff is. Actually, it's wonderful.
So if you can see now, most of the abdomen is pretty much empty. We've taken all the organs that are there. I am going to ask Laura to step back while I take the scent glands out.
Now, when you say the scent glands out here, that sounds exceptionally smelly.
They are the smelliest part if ruptured. We do our very best not to rupture them when they come out.
My fingers are crossed, Emma. My fingers are crossed, I tell you.
I will do my best.
Yeah, best to be out of the firing line.
I was going to say, is this something that could spray in this direction
you are
as well?
in the firing line, so
do you mind? I'm just going to step to one side here with Liz and have a chat to her.
And maybe just a quick thing before Emma starts. Emma took the testes out and we've trimmed all of those, and we've got these other tubes called the vas deferens, and we have actually found one cyst on here.
Oh, I can see that. Oh, that's tiny,
it's like a little kind of bubble thing that's appeared there.
So what does that mean? What's the relevance of finding a cyst? I found a cyst. What does that mean?
It's interesting. There's a researcher in Sweden did a study a few years ago and showed that these cysts on the vas deferens are potentially associated with chemical contaminants. So that's something that we've only relatively recently started recording. It's always the way, the thing that you didn't notice or didn't record before is the thing that you then think, damn, if only I'd been recording that since the beginning.
Foresight, eh?
So we are now recording those and as well. But it may be linked to chemicals that are affecting reproductive health.
That's so interesting.
So
we're just gonna stand aside. Don't worry about us. Don't worry about us.
So you can start to see them emerging here. The two lumps.
Oh, they're quite. Do you know what? They're bigger than I thought they'd be.
Yeah, they're nice and full.
Wow, they are big. They're bigger than the testes, aren't they?
Yeah.
Goodness me.
Yeah, you can really see there the two.
Yeah.
I'm so glad you moved me out of the way now, I can tell you.
There's a lot of bruising down there, which makes Emma's job a little bit harder because there's a lot of swelling and so on. But she'll try and get them out intact without rupturing them. There we go.
That's a skill, isn't it? Wow, that is a skill.
So there's this pair of glands that sit either side of the anus. So you can see that's where the anus was, and the glands sit either side of that. And when they defecate, so when they deposit their spraint, the faeces, they're actually putting down these scent markers, which we think that the otters are using in communication.
I was going to say, because if I see scent marking or sprints when I'm walking around waterways, it's normally on quite a prominent log or something like that in the water, and you find a sprint mark in there. So they're almost leaving a calling card, it's my territory. Yeah, that's it. So another otter can come along, smell it, and there we go. It's fascinating, isn't it?
I'm now going to go into the thorax, which is again another kind of elbow.
So what is best? Do you want me to stand this side out of the way?
That's fine. So I'm just going to cut slightly, not right down the middle, because it's quite hard to get right down there. It's a lot easier to go just to one side.
There's a line of cartilage that Emma will find, which makes it much easier to cut.
Right.
So while she opens that up, I can tell you a little bit more about the scent communication. So that's something else that we've done some research on. So Eleanor Keane, who is a postdoc with the project, she did some brilliant research linking the chemical components that are in the scent signal to other characteristics that we record during the post-mortem. So for example, the sex, the age class, the reproductive status, and so on, so that we can see what it is that those otters are communicating, what messages they're
exactly. Wow. Reading otter scent glands, how incredible.
So it's a really fascinating area of research.
I'm just looking
at
that.
One half is opened, yes.
Goodness me.
So I've just cut down on each side of the diaphragm here. You can really see this is the tissue that we mentioned before that was intact. Obviously it isn't anymore.
So the diaphragm, that's helping to pump the lungs, helping the air to get through. So that's such a neat cut there. So there's a layer of soft cartilage then in between the ribs, which is, as long as you can get that and cut down that, it helps you to get it through the rib cage. Is that on both sides? I guess?
And again the other when halfway down the ribs there's also an area of weakness that we cut along.
Right.
It's quite hard
If
to
Emma was cutting directly onto the bones, all she'd be doing is blunting their scalpel
Absolutely.
and fighting
Yeah.
with it and
Imagine,
struggling.
I can't cut through bone. I'm
No.
not.
Well, no,
No.
yeah, I should imagine. I should imagine the risk of cutting yourself as well.
Yeah,
With the
yeah.
scalpel. So this
But
is.
yeah, if you know where to cut, then it's
and that's
relatively
just
straightforward.
opened up
yeah.
the rib cage,
Yeah.
hasn't it?
Yeah.
Completely.
So we'll do a thoracic overview as we did with the abdomen. So it all appears pretty much in place. That's kind of the heart is where it should be. It's not randomly on the right side. It's on the left and kind of the central thorax. There is still a lot of free blood in the thorax, as there was in the abdomen,
Yeah.
which again is probably due to the impact. We'll make a note that these lungs are quite mottled in their colouring. If you can see
Yes,
that.
I can see
I'll
that.
cut
Yeah.
one out for you.
So is that unusual to have that mottling on the lungs?
You quite
Oh, that's
often
really see it
see
now.
There you go.
in these road-killed individuals, which I think this one we're pretty
Yeah.
confident is, can see quite a lot of bleeding into the lungs,
Oh, of course.
and then with time after death, that will really discolour and darken, and
Of course.
so it's almost certainly not any sign of illness. We would be kind of really feeling it's the texture really that tells us a lot. It's a really sort of rubbery feel to it. I can see there's been a rupture there.
Can I get some gloves on and have a
There's
feel of this, Liz?
some
Hold
spare
on.
gloves
We've
there.
got to find some large gloves. Hold on. I just it's time
Yeah,
to get
feel
messy.
free.
I think. Let's have a look at this. Because it's one thing you telling me what it feels
Yeah.
like. I really want to try and understand exactly
Yeah.
what it is you're trying to find there.
It's very like a sponge for
Yeah.
people at home who obviously can't feel it today.
Feel
Feel
a
a wet
sponge.
sponge. That's
Okay.
kind of
So what? I'm going to pick this up now. Oh, it's so much lighter, isn't it, than you
Yeah.
expect it to be? Goodness me, you can hardly feel it on your hands.
If you press
Wow.
a little bit, you can actually feel those
Little
air
spare
pockets.
and so on.
Yeah, absolutely.
So if there
Oh
was,
yes, no, yeah, you can really
Yeah.
feel it, can't you?
yeah.
Wow, that's quite incredible. I can't believe how light that is. It's like half the weight of say jelly or something.
Yeah.
Half the weight of it. It's
Yeah.
very, very, very light. Very light.
We don't actually normally weigh or retain the lungs. We just make a note of any kind of huge abnormalities.
Yeah, give them a
Um,
check over.
The things that we do weigh in the thorax are the heart and this little organ, the thymus, which is quite hard to see
Oh, yeah.
before I take out. It sits just above the heart here. If I tilt the thorax over a bit, it's just along
See it, yeah,
here.
just on there. Wow.
And I'll just take that out. So you can-
Again, because there's so much bleeding in this animal, because it's been hit, there's just bleeding everywhere. Everything kind of ends up looking the same colour, just this dark. But if you had an animal that was, had died without being hit like that, you'd see much more differentiation in the colour of the tissue. So it's actually a much easier job for the pathologist to kind of tell what's what and see where they're going. But it's quite hard when it's in this.
I'm really appreciating just how skilled you girls are, because you're
Yeah.
pulling stuff out which is just
I have
unbelievable.
cut quite a lot of spare tissue around there, but you can see this little bit here.
Oh yeah,
This is the
yeah.
thymus. This is just a little bit of.
Blood vessel.
I'll let Laura.
Again, when you're looking at that amongst all of that
Yeah,
sort of goo and
yeah.
mess.
It is very much, you learn the different textures as much as
Okay.
anything.
Yeah, that makes sense.
Um,
Yeah, I understand.
Textures, colours, but colours can be a
Yeah.
bit hard when
Yeah.
it is a lot of
Yeah.
bruising and a lot of blood everywhere.
And the thymus is very, very variable. Sometimes in the older animals, and this one is, seems to be a mature male, the thymus can become completely regressed, so we don't find it at all. But in younger animals, it tends to be larger. So it's the sort of teenage otters
Okay.
have a larger thymus because it's producing all sorts of hormones and so on.
So I know how long is a piece of string. This is a very vague and subjective question, but any idea at all how old this animal could be? I know it's probably guesswork, isn't it?
Yeah.
Or a range, maybe.
I'd give you a range. So with the things that we're looking at, obviously there's the overall size.
Mhm.
We can see that he's adult size. He's quite a large adult. The baculum is
was
giving us a
quite
clue
big, yeah.
that was over 70 mils, so again indicating that he's larger. I'd want to have a look at the teeth as well. So I think Emma examined those earlier, and we'd be looking for signs of wear.
Can I get in there as well?
Yeah,
Can I have a look
sure.
at these
Yeah,
teeth?
Sure. You can
Oh,
see.
goodness me.
You can see that when we look at the teeth of really old otters, they can be really worn down, and these lower incisors can be quite sort of stubby, worn right to the gum
Okay.
line. Whereas
But
this
that doesn't
one,
look like that
no,
to me, Liz. That looks...
no, not at all. It's not, and it'll vary depending what they've been feeding on. In some areas, if they're eating a lot of crabs, crayfish, that kind of thing,
okay, yeah, so
they
it's
might
diet dependent.
wear down even in quite young otters. So all of these things are not necessarily reliable on their own, but there's all these different things that we're looking at.
Absolutely.
So, yeah, in terms of giving you a number, we know that in the wild, they're certainly known to live up to 8 years old.
Right.
But if he was at that end of the range, I'd be expecting more tooth wear. So I'd take a wild punt and say maybe somewhere between 3 and 5.
Yeah, okay.
But yeah,
Brilliant.
who
Wow,
knows?
it's so complicated.
Without getting that tooth sectioned.
Yeah, exactly. Yeah, you could be
get
more
a lot
specific by doing that, but just guessing at this point, it's
Reshuffle.
Yep, I'm going to take
3
the
to
heart
5
out
years
next.
old.
So it's a pretty intact heart. I can't, although I'll cut it out to see if there are any.
I find it remarkable. You said that traumatically sometimes that will migrate all the way down into the abdomen.
Yeah, we can find the
Wow.
heart, amongst the kidneys, and severely fragmented sometimes. This one appears pretty intact, and you can have a feel down there. You can feel all the different.
Shall I do that?
chambers.
Shall I have a
Yeah,
look?
do it.
Okay.
And we'll be able to open it up as well to show you a little look inside.
Okay, so the different chambers.
Yeah.
So similar to the human heart with the atrium ventricles as well.
Can I say something? The walls are really thick
Yeah.
on that
Yeah.
compared to
Laura, do you want
other
to
mammals.
cut it across?
Yeah,
and
so if I
then
cut it
That's
open.
huge. Yeah.
Which we do normally actually cut it open to see if there is any abnormalities inside.
Okay.
So yeah, it can be a bit slippy, so
Okay.
bear with me.
right across.
Yeah.
Yeah.
Take your time.
So it is very similar to a human heart.
Yeah.
So it's quite interesting to see.
It just looks really
The thickness
powerful,
of the walls
isn't it? The thickness of
is
the walls.
quite
I noticed.
unusual.
Yeah.
And you will often get the thicker side is because it needs to pump the blood all over the body. That's why it is so thick, because it needs so much power.
That is a
So
powerful
you can
heart.
see,
Yeah.
yeah.
Wow.
And you can kind of see the valves in there, not very well, but
I'm absolutely amazed at how thick the walls are on the heart
Yeah.
of the otter. That is remarkable.
and you can see that it is thicker on that side
Yeah.
compared to this side as well.
Wow. This is a great learning experience. I'm learning so much myself here. Just a thought as well, if you guys at home, if we're generating questions for you and we're not answering those questions here now, don't forget we've got the girls upstairs who are still answering your questions on Twitter, or hashtag AskOtter2018 on Facebook, and at Otter Project UK, and also by email if you want to send your questions in, otters@cardiff.ac.uk. Do keep the girls answering the questions upstairs, give them something to do. So another big shout out there to Neil, Lorna, Amy, and Juliet, isn't it? Juliet's the other one,
Yes.
all working away hard upstairs.
Yes, Juliet's doing work on the contaminants, so that those hair samples that we were talking about, she's doing some work testing for pesticides in hair, and also trying to develop analyses looking at other contaminants in the liver.
Right, okay. Things like pyrethroids and all sorts of things like
Yeah,
that. And
so perfluorinated substances,
Yeah.
and a whole range of pesticides as well. So yeah,
interesting.
lots of different contaminants.
So the final thing I'll be taking from the thorax is the rib here.
Yes.
We take the rib from the right side and we count the 7th rib up, and we'll just take that, and that will go in the bone bag with the leg and skull later, and as well as the baculum.
So it's really keeping that consistency. It's not even a rib, it's the 7th rib down. Wow, okay.
Yeah, so we just make sure that all the excess tissue which is mostly muscle from around the ribs are trimmed off, which can be a bit tricky.
Is that because you're weighing it as well, and you want to keep consistency again?
The rib, it's mostly we clean up all the bones, and it helps out those who are archiving those bones up in Scotland. The more we can strip off at post-mortem, the less they have to strip later.
Right.
So, yeah.
So while Laura does that, I'm going to start cutting into the throat. Yeah.
Can I get behind you for that? Can I have a?
Of course, come
I'm
round.
being terribly nosy, aren't I?
I'm going to be taking out the thyroids, which are the hardest part to take out. So I apologise if it takes
okay,
a while. You will be able to see the trachea as I cut in. If you can start.
Oh, you can see that. Look at that.
Yeah.
It's got trachea always reminds me of sort of like a long. It's a long pipe, isn't it? But you can see
it's like the hose of a vacuum
Yes,
cleaner.
that's what I was thinking. Yeah.
So if I tilt it, you can kind of see it starting to emerge there. And I'm then going to, bear with me, either side, make a slight cut under the trachea.
Mm-hmm.
And pull up this tiny bit of fat, and I don't know if you can see the dark colouring there.
And it really is a tiny piece of fat.
Yeah.
I've got to say, there is a huge amount of skill being shown here, because this is really incredibly difficult, and you're making it look very easy.
And again, I have to thank Vic Simpson for that one, because I never used to be able to find the thyroid glands at all, and he gave me that tip about looking for this little line of fat that follows a nerve. You find the end of the nerve, and then you can pull that up, and the thyroid will emerge.
It's tiny, Liz.
Yeah, so I've taken quite a bit of tissue off with that, which Laura will trim in a minute. But you can just see there how the texture changes, how there's a slight gap between the organ.
Oh no, I can see it, there.
And this is just excess tissue, so if I cut this bit off.
See, it really is texture is king here, isn't it?
The texture of muscle is very different to the texture of the glands, and that's what we're...
Look at that, I'm just going to put my finger next to that so you can see how utterly minute that is. And finding that to cut out, I mean, that is incredibly skillful. I am so impressed.
And it is with that little bit of fat that you did. I remember, the very first time we did that post-mortem, you made it key that we have to remember to find that little bit.
It's really tricky.
So what's so important about the thyroid glands? What are we
Again,
going to find here?
it's as with most of the other organs that we're taking, we weigh them when we're looking for abnormalities and inconsistencies. So obviously if there was any sort of growth, lumps and so on, that would be more obvious, but we'll check the weight as well to see if there's any abnormality there. And we'd be looking later when we come on to do that sort of statistical analysis, can we see any associations between, for example, the relative weight of the thyroid gland and chemical contaminants that we're measuring in, say, the liver or the hair, and it's all of these things that can indicate the health.
So I guess with this many samples off this many individual otters as well, you're looking in the thousands, and all these different samples, you're looking for sort of correlation between sort of inflamed thyroids and this and that, and all of the what are the indications to disease, to contamination, to all sorts of things. So interesting.
So we don't usually take the trachea out, but I thought I'd take it out so you can have a feel of the texture at home.
Fascinating, isn't it?
And this runs all the way down.
This is not something you do every day. Oh, look at that, all the way down.
All the way down there. I'm just going to cut this top bit out so you can have a feel of the...
Wow, look at that. You can see it really is stretchy as well.
You're going to get the lungs as well, Emma, to show.
Yeah.
Look at that. And you can't compress it, I notice. It's sort of
so
difficult to compress.
Emma's taking the lungs out. I can show you a little bit more.
Oh, go for it.
You can kind of see that you've got these rings of cartilage.
Yeah.
And then at the very back, it's not actually like a full tube. It's kind of more of a C shape.
Yeah.
So that allows the trachea to expand while the otter is breathing in. So very similar to our trachea as well.
So this is kind of the other end of the trachea. It's quite hard to show you, but the lungs, so this trachea here leads all the way down.
That bit that we cut off.
This is the extra bit that should be there somewhere.
Again. So that
There's one we prepared earlier.
that sits in the thorax, obviously there. And then that trachea will run all the way up to the throat, to the mouth, where the air obviously goes in.
and we get these 2 lobes on the left and 4 lobes on the right with the lungs in the otter.
Yeah. You can see it's a bit, obviously not quite in place.
Yeah.
And you can really see that mottling now, which is it's just where blood it's
blood's pooled
in there. Yeah.
And you can actually see the difference between the throat, which hasn't had any massive trauma, compared to the colour of
all this
the
kind
rest
It's
of
a
the
lot
otter.
lighter, isn't it?
Yeah.
Yes, it's very, very dark down here, very, very pale here.
Like more of a vibrant kind of red when it is very fresh.
It does actually make it easier to take the thyroids out. So I'm quite glad it hasn't been hitting the throat.
Yes, when they're very bruised and swollen as well, it does. It makes it so hard to find them.
I take it that some of them can be, obviously, if they come into conflict, in traffic, that you can crush the back of the skull, or head impacts. That can just darken considerably. You wouldn't see that.
Yeah, quite commonly we see crushed skulls, broken legs, all sorts.
In that case, we wouldn't take the whole skull out, we'd just take a section of the jaw.
Of course, yeah.
But in this case, I'm going to take the whole skull out.
Right, wow.
So, bear with me while I peel back.
Take a little while to get that all peeled
Wow.
away. When we look at the skull, there's a number of things that we can look at. So we've talked about the teeth, because we can extract the canine and section it. The skull itself, some of the things that we're looking at, for example, are what we call cranial sutures. So these are where the bones join together, and in very young individuals we see this kind of wriggly line where they're not quite fused.
Yes, yeah.
And then as they get older, that thickens and fuses together. So again, we can use that as another age indicator. Another thing that we see is in the older animals, particularly the older males, that have really, really strong biting muscles, we see a kind of a ridge on the back of the skull, which is where all the muscles will attach.
All the jaw muscles attached, so they've got stronger jaws. Wow, okay.
Yeah, so
I'm used to seeing that in
see
things like badgers and whatnot, But
it's not as prominent in an otter as it is on some of the
Is it sagittal
skulls.
crest?
Yes,
is that right?
right.
Yeah. Okay.
Yeah.
So I didn't realise that otters actually had those as well.
They
That's
do,
incredible.
Yeah.
So I'm just cutting down. I've taken off the pelt around the lower jaw. You would normally see the tongue here, but obviously
Of course, you
that
took
out
that.
earlier.
Saw the tongue coming out. Well, that's remarkable.
You can see there's a little bit of pelts left on.
Don't forget, if you've got questions at home at the moment, don't forget you can still call into us now. You can send us an email on otters@cardiff.ac.uk. You've also got Twitter and Facebook. You can get hold of us on Facebook at Otter Project UK or on Twitter at Ask Otter 2018, and all of that information should be coming up on your screen now. Wow, the whole top of the skin, the pelt now is off, and the skull is considerably smaller than I thought it was going to be, doesn't it?
The amount of muscle.
Huge amount of muscle on the head,
Yeah. Those jaws are incredibly strong. So,
I would not have believed that. I really was expecting a larger skull proportionally for what we'll see. So that does really go to show the enormous amounts of muscle around that jaw.
And at this point you can really see the teeth as well, and at this moment otters don't look as cute, they look a bit more scary.
We'll be able to see how when the jaw moves, you can actually see this muscle from the top of the head moving.
Goodness, look at that. Can you see that at home actually? If you just move that, look at that. You see just how much muscle is involved in making sure those jaws
these thick
close
pads of
effectively.
muscle are coming up off the top of the skull there.
Really is incredible. That's why the head looks so large.
I will try and take as much muscle off as possible just for the National Museum of Scotland, so they don't have to clean as much off. But there will be some left.
Because cleaning skulls takes a few months, doesn't it?
So one of the things that we haven't talked about is the fact that so many of these animals are being killed on the roads, and one of the other things, not so much from our post-mortem research, but simply from that information about where they've been killed, we can collate those location data together and share that with organisations like the councils, the wildlife trusts and so on. We can look for hotspots of mortalities.
I was going to say, is there any chance then, if you've got that type of data and you can, there is a specific road and we are getting conflicts on that road every single year, there's high mortality, is there any mitigation that can be put in place to try and alleviate that conflict?
Yeah, absolutely. So there have been quite a large number of places now, we've had mitigation fitted on roads, things like ledges under the bridges, so that
Yeah.
when they're sort of coming along the riverbank, instead of having to pop up and over and cross a road, they can just carry on along a ledge underneath the bridge, so they don't come into conflict.
And they're
with
relatively
traffic.
simple solutions,
Yeah.
aren't they? These are not
Yeah.
really expensive things.
Not at all. No.
So, no, it's fantastic. And are
It
there
can be.
organisations that, is that down to the council to implement those, or are there organisations that are assisting in sort of alleviating the conflict of cars and otters?
It really varies from place to place, and depending on who's got funding where and so on. So yeah, it's very variable.
Okay.
Just
Whoa,
finish
out
that.
comes the skull. That just popped
Yeah,
out, didn't
it's
it?
just come out, so it's still got quite a bit of muscle tissue around it.
I think we should definitely get a good shot of that for the people at home
Yeah.
as well, just so they can see,
So,
because that's quite remarkable. It's not a sight you see every day. An otter skull just as it's come out.
Would you like a hold
From the
actually?
animal. Can I hold that? Oh goodness me! Okay, no, I definitely don't get to hold. Look at that! I am utterly amazed by the volume of muscle here. Just how much muscle? Because the muscle is coming right across the top here,
Yeah.
and it's the power of those jaws. I mean, you can see just how powerful those jaws are. There was no way from looking at the animal that I was able to be able to recognise just how powerful those jaws are. And now, during the autopsy, we've got a window on the inside of this animal, haven't we? The inside of the skull, and it's just utterly remarkable. Those jaws, something
you
I'll
can see as well,
never
both
forget.
of the eyes are present here.
Yeah,
It's probably quite normal because the skull hasn't been hit by a car, but often with the crushed skull we have one eye protruding, one eye missing, which is quite sad to see.
and again, I'm just feeling around here. The skull feels pretty solid.
Yeah.
Yeah.
It's a solid skull anyway, isn't it? It's a real solid skull. Never mind about the muscle.
Can you feel the crest on that one? Has it got much of
There is a tiny crest,
tiny
Liz.
one?
I don't know what.
Yeah,
I mean,
let's have a-
that's what I think is a crest. Is that correct?
Yeah. It's not. It would look more prominent once we've cleaned it up and stripped all the muscle off, but it's not particularly large. I've certainly
Wow.
seen larger ones.
They
Big
get
to me.
bigger.
Just big to me. And that crest is there, so all
Yeah.
that muscle tissue can sort of, physically can attach to the top of the skull there
Yeah.
and power those jaws. That is
Yeah.
utterly remarkable.
And the pelt looks a bit sad now.
It does, doesn't it?
With no jaw left, it's
But
just a bit
I think if I can just show again, because this I find remarkable.
back
I'm
in.
just looking at the size. If we just have a look at the, if you just look at the pelt and you're looking at the head there compared to the skull, I mean the skull just looks as if it didn't come from this particular sample.
Yeah.
It's just so small, the skull compared to that. And as you say, if you're going back, can I have a look in there? Look at the muscle, the muscle powering those jaws. That is utterly phenomenal.
Yeah, I've got these two big sections. I've also just seen on my hand I have a little bit of white liquid. That's
Yeah.
actually brain material.
Oh right,
So
okay.
sometimes when we're cutting them completely off, you might be lucky enough to have some on you as well.
Oh,
you do
right.
get bits of brain matter kind of seeping through occasionally.
Otter brain matter. All
Yeah.
right, okay. Well, look, I'm definitely in the way here. That is, for me, that was the biggest take home so far. I've been utterly amazed at that. So incredibly visual.
So while you've got your hands dirty, I
All
don't know
right.
if you want to have a closer look at the pelt itself,
Yeah.
because it's an amazing pelt. If I pull it back there, you can see.
You can't actually get down to the
density.
skin. Yeah, no,
No.
the density. So this looks
you
like
can't see skin at all.
no,
It's just,
you absolutely can't. So it's almost like it's. I'm going to sound silly now. It looks like it's two layers, and there's like a finer layer underneath there.
yeah, the underfur is really, really dense, insulating layer, and then the longer hairs on top of the guard hairs.
They are guard hairs. I was going to say
really
guard hairs. Okay.
sleek.
So that would be what we'd normally.
Will be helping with kind of shedding the water.
Okay.
Yeah.
But you can see just how incredibly insulating that would be as well.
If we can get Laura to just run through our checklist.
Yes.
Start
Yes,
and make sure we've done
We have
everything.
just had a quick run through, but
Yeah.
Always
Okay.
better to do it.
yeah. So we got the adrenals, but we only
Wonderful.
retained left. We got the kidneys. We didn't find any kidney stones. We got the spleen. And we've got all the liver samples, thymus, heart, thyroid, baculum, testes, vibrisine. There was no ectoparasites. We got the tongue, pelt section, gallbladder, blood, scent glands, all muscle samples, rib, leg, and the skull.
Brilliant.
Yeah.
So when you were saying ectoparasites there, where in particular would you see the ectoparasites? So these are not the ones like you said that, I think you said the gallbladder might have some parasites in endoparasites, but the ectoparasites, things like ticks, yeah?
So that's what we were checking.
Where
at
in
the
particular
beginning.
would you look
I
for
just fold
those?
this over. It's hard now without the skull,
Do you want me to
but
like-
generally between the shoulder blades here,
Okay.
and then in and around the ears.
It's the areas where they're less able to
I was
move.
going to say it's the hard to reach areas, isn't
Yeah.
it? It's
That's where we
obvious,
tend
really.
to find them.
So they're usually quite obvious. If they're large ticks, you'll see kind of a black or white kind of blob.
Yeah.
But they can be really tiny, almost specks in the ears. But again, like I said at the start, this one seems to be in pretty good condition.
And sometimes even
for its age.
yeah, and sometimes even inside the ears, we have to have a double check in there as well.
Yeah. So I think we've taken all that we can from this otter.
Yeah, a huge thanks to the Cardiff University Otter Project. Also, we should say a massive thank you to our sponsors today as well, the
100%.
International Otter Survival Fund there that have helped fund this and allow you to see it at home today. Don't forget, it's your last chance now. Quickly get in your questions. You can still contact the girls upstairs. They're working away hard you can send an email on otters@cardiff.ac.uk. We've got also Facebook there at the Otter Project UK, and we've also got Twitter, which is hashtag AskOtter2018. But from all of us here today, we'd just like to say a big thank you for tuning in, and we hope that you got something out of that as well. I know I did. So from everyone here at the Cardiff University Otter Project, it's goodbye. Thank you very much.
Thank
Goodbye.
you.